discussion is the intercellular junction. So as you know, epithelial cells as well as other cells, they are connected to each other. They join to each other by certain junctions. So what is the junctions? They are the communication between the cell, or they are the means of attachment between the cells, or they are the sites of attachment or communications. And morphologically and functionally, there are three types of junction: tight junction, adhering junction, and gap junction. Cells communicate each other by attachment or the junctions. Three types of junctions are there. One is tight junction, adhering, and gap junction. So tight junction, the name indicates is very tight, attached very closely. So that's why they are called as, they are unique to the epithelial cells and consist of irregularly anastomosing ridges that seal the neighboring cells in a belt-like fashion. At these ridges, integral membrane proteins, adjacent cell membranes fuse across the intercellular space. So they limit the movement of enzyme from one cell surface to the other. So that is about the tight junction. They are very unique, and they are unique to the epithelial cells only. They shield the cells in belt-like fashion. They seal, they shield the cell in belt-like fashion and maintain the cell polarity by restriction of the movement of the enzyme from one surface to the another. So adhering junction. So adhering junction, these are three types. Three types of adhering junction. That is called as bell desmosome, spot desmosome, and hemidesmosome. Bell desmosome, spot desmosome, and hemidesmosome. So bell desmosome, it is also known as zonular adherence. It is located always below the tight junction and surrounds the epithelial cell in bell-like fashion. Spot desmosome, that is a disc-like structure. Disc-like structure, approximately 200 to 400 nanometer in diameters. And there is present within the intercellular space. And hemidesmosome is about the half of the desmosome. So type is the one number, adhering junctions. The second broad category, adhering of three types, that is bell desmosome, spot desmosome, and hemidesmosome. And the last type of junction, the last categories of junction is the gap junction. It's very important. The gap junction bridges the connection formed by 6 integral membrane proteins of the opposite membranes, which are complementary and interlocked. And it is the gap junctions that allow the direct passage of ions and small molecules from one cell to another cell. That is always a question. The junctions which allows the movement of ions and small molecules from the cell to cell. Okay? Or they will write the junction which allows the direct passage of ions and small molecules from cell to cell is known as gap junction. So they are absent in the skeletal muscle, spermatozoa, as well as the circulating blood cell. So here the location of the tight junction, unique to the epithelial cells, then the adhering junction, okay? And then you will find a gap junction or a nexus. These gap junctions which permits the passage of the ions on the molecules from one cell to another. The same the pixelatic view, the tight junction, adhering junction, and the gap junctions. And hemidesmosome, normally you will find this bind the epithelial cells with their basal lamina. Then next the junctions, as I have told you, we will discuss about the specialization of free surface. Free surface means the apical surface. So have you seen the slide?
Yes, sir.
Yes, sir.
So suppose this is a cell. The slide itself, you see the PowerPoint slide itself, a cell. So in the cell, the top, this surface will be called as the apical or the free surfaces. Then the down this you will find that it is called as the basal surface. This is the basal surface, and on either sides you will find that is a two lateral surface. This surface and this surface, the lateral surfaces. This is about the surface of the cell. So now we are going to talk about this surface, the free surface or the apical surface. There is certain modification. There is certain modification of the free surface of the cell. So now what are those modifications of the free surface or apical surface? Always remember, specialization is there means that is in the free surface or apical surface. Okay, when there is modification on the lateral surface, so lateral surface modification means that what we have studied that gives the cell junction, that's for the cell communication. Clear? So if somebody asks you what are the modification of the free surface of the cells, the free surface modification will give like cilia, flagella, microvilli, stereocilia. But when there is modification or specialization of the lateral surface of the cell, that will give you the cell junction or the communication. Now first see what is the cilia. So cilia is about 0.2 micrometer in diameter, so between 5 to 15 micrometer in length. If you see on the electron microscope, you will find that cilia consists of an axoneme surrounded by plasma membrane. It consists of axoneme surrounded by plasma membrane. But what is this axoneme? So if you see the cilia you will find in our nasal epithelium or many locations in the body. So if you see at the electron microscopic level, you will find that it consists of axoneme surrounded by plasma membrane. Now what is this axoneme? Axoneme is composed of 9 doublets of microtubules around 2 central microtubules. And cilia are rigid in the effective stalk when propelling mucus and become flexible during the return. Now what is the flagella? A single long cilium is referred to as flagellum. You will find in the mammalian spermatozoa. So cilia means it consists of axoneme surrounded by the plasma membrane. Now what is flagella? The single long cilia. It is called as the flagella. You see? Now what is the microvilli? So in cells, especially intestinal epithelial cells, whose principal function is absorption, those cells which main function is absorption, free surface has varying number of cytoplasmic evagination. This is called as microvilli. Clear? You get the point. Cilia means it consists of axoneme surrounded by plasma membrane. If it is single cilia, that is called as flagella. If it is a cytoplasmic evagination of the free surface, especially in the intestinal cells, that is called as microvilli. And you see, especially in the organ of hearing, receptor cell and vestibular sensory receptor, they have long rigid microvilli that are called as stereocilia. So what is because stereocilia and microvilli, they are similar, because it is the stereocilia means it is long rigid microvilli. But how they differentiate? These stereocilia, they are narrower at the base than at the tip. That means at the top part, okay, they are narrower than at the tip in case of stereocilia. So here this is you can see the cilia. So I'm telling cilia consists of the axoneme. So this entire structure, you can say, this structure is called as the axoneme and this is the plasma membrane. So cilia consists of the axoneme, and axoneme is surrounded by the plasma membrane. Now if you see the axoneme, so what you will find in the axoneme, you will find 2 central microtubule. Here you can see 2 central microtubule which is surrounded by 9 doublets of microtubule. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 doublets of microtubule will be surrounding the 2 central microtubule. That is a structure of cilia under electron microscope. And if you see these 9 doublets of microtubule, so you can see one microtubule is complete, another is not completed. And these 2 microtubules are connected by the dynein side arms. Okay? And all these microtubules are connected to the central microtubule by the radial spoke. I'm not going to the details, but I'm just telling, but you must know it about what is cilia. It consists of axoneme surrounded by plasma membrane. Axoneme means 2 central microtubule surrounded by 9 doublets of microtubules. Here you can see the view of the cilia, microvilli and stereocilia. Here it is cilia. Okay, this is stereocilia and this is the microvilli, evagination of the cytoplasmic surfaces. Now, what is the centrioles? Do you have any idea about centrioles? All animal cells have 2 centrioles. So what is the function of these centrioles? So they help the cell doing cell divisions. Centrioles, they help the cell during cell division. They work during the process of mitosis as well as in meiosis. Usually present near the nucleus, but they are not visible when the cell is not dividing. That means during the cell division only, during the process of cell division only centrioles are visible. They are not visible when the cell is not dividing. And what is the function of the centrioles? The main, you see, helps in organizing the mitotic spindle and complete the process of cytokinesis. That means formation of the two cells. The main function is the centriole help in mitotic spindle formation. Centrioles are believed to be necessary for the formation of mitotic spindle in animal cells, although several recent types of research have explained that the cells which does not have a centriole surgically removed to the cell functions without it in GI level of interphase and can be formed later in vivo. That is different. Present research is showing that without centriole also cell can functions. Now, in short about the functions of the centriole. So in spite of being devoid of DNA, centrioles are capable of forming new centrioles. They can be transformed into basal bodies. The basal bodies give rise to flagella and cilia. They help in cell division by forming microtubule organizing centers out of the two centriole distal centrioles from the tail or the axial filaments. Is the slide is visible?
Yes, sir.
Yeah.
Yes, sir.
So now we'll discuss about the cell divisions, okay, and especially the eukaryotic cell cycles. So this eukaryotic cell cycle is divided into two major phases. Okay. So the first and the second. The first, you see, what in the two major here you will find in the first phase. In the first phase there is three stages. This you remember two phases, and in each phases there is three stages and four stages. So first phase means here what you will find that is called as the cell cycle, the G1, S and G2 phase. The first phases means the G1, S, synthetic and G2. These are the three stages of the first phase. And the second phase, that is the mitosis. The mitosis having four stages plus cytokinesis, where the nuclear division and division of cytoplasm takes place. So total cell cycle, it consists of two major phases, first phase and the second phase. In first phase, there are three stages, include G1, S and G2. And the second phases, there are four stages, okay, that includes the stage of the mitosis. So you see that non-dividing state with the three substages. In here, what you will find, a cell grows in the interface, cell grows in size, organelles replicate. And you know that the first phase that means G1, S and the G2 stage. I just showed you, okay, G1, S and G2. Now what will happen to the G1? Cell grows in size, organelles replicated. S phase, replication of DNA, synthesis of proteins associated with DNA. And G2 stage, synthesis of proteins associated with mitosis. These are the three phases, three stages of the interphase. Okay? G1, S and the G2. Now let us come to the second phase, that is mitosis. What is the mitosis? Division of the somatic cells, that is a non-reproductive cells in eukaryotic organisms. A single cell is divided into two identical daughter cells. Daughter cell have the same number of chromosomes as does the parent cells. Now what are the stages of the mitosis? As you know, So here you see this packing for the move, the cell, they are ready for the mitosis. When the cell is not divided, DNA molecules are extended, uncondensed from chromatin, only replicate. Cell is preparing for division. DNA molecule condensed from the chromosome prior to the division. Chromosome are in single molecule of DNA. This is just before the starting of the mitosis. So this mitosis is having 4 subphases. That is, first is called as the prophase, second the metaphase, third anaphase, fourth telophase. Then followed by the telophase, that is the cytokinesis, when you will find two complete division of the cell nucleus. Okay. So prophase, what will happen? A major event in the prophase. So chromosome will condense of the chromosome. Then the spindle fibers is forming. This is spindle fibers form, and spindle fibers are specialized microtubules radiating out of the centrioles. And these chromosomes are captured by the spindle. These are the 3 major events you can see. Condense of the chromosome, formation of the spindle fibers, and chromosome are captured by spindle. Condense of chromosome, spindle fiber formations, and chromosome captured by spindle you can see in the prophase. Here, prophase stages. Metaphase. So what is the major event in the metaphase? Chromosome align along the equator of the cell. You can see chromosomes. They are aligned. Chromosomes are aligned along the equator of the cell with one kinetomer facing each pole. Then in anaphase, so what happened in anaphase? The chromatids, they are separate. In anaphase, sister chromatids they separate. Spindle fibers are attached to the kinetogears, shortens and pull chromatid towards the poles. Chromatids are pulled towards the pole. Three spindle fiber lengthen and push pole cell apart. These fibers, spindle fibers, they become lengthen and push the cell apart on either side. Okay. So main here in the anaphase is separation of the sister chromatid, and spindle fibers lengthen of the spindle fibers and poles and push the poles cell apart. And in the telophase, what will happen? So these spindle fibers they disintegrate. Spindle fibers disintegrate. So what will happen? The nuclear envelope. Once the spindle fiber disintegrates, the nuclear envelopes forms around both groups of chromosomes. Chromosomes, they revert to their extended state. And then cytokinesis occur and closing each daughter cell into a separate cell. So after the telophase, start the cytokinesis. So animal cells undergo cytokinesis through the formation of a cleavage furrow. You can see the cleavage furrow, a ring of microtubules contact, pinching the cell into half, thereby formation of the two individual cells by the process of mitosis. This is a schematic diagrams of the mitotic cell division. Okay. Do you have any point here? So then I have some question. So regarding the meiosis or the gametogenesis, no, while we study the embryology, there we will discuss, because in the cell division we are here discussing mainly the mitosis. So gametogenesis again you will study while you deal with the what is called the embryology. So you will study the meiosis division here, okay, not here again. So now I have certain points for you. So please response to it. Please respond to it. Can anybody? Huh?
Robot.
Okay, good.
D.
Energy transaction, it will be there. Otherwise, how the exocytosis will take place? Exocytosis, endocytosis, then you the energy. This is the number D, assisting in chromosome segregation. Not the function of plasma membrane. Number D, assisting in chromosome segregation.
D.
D.
Yes, it is a D. Plasma membrane is bilayer of phospholipids. Bilayers of phospholipids with the protein layers as well as carbohydrates you will find attached to its external surface. Okay, the correct answer is D. It is not the bilayers of proteins. It is not consist of equal numbers of phospholipids and proteins, no equal numbers of phospholipids and proteins, and it is not composed of one layer of phospholipids. It is the answer is D. On the external surface, yes? Yes. The main role of carbohydrates in the plasma membrane is recognition. The polysaccharide layers, which is known as the glycocalyx, you remember? You may find a polysaccharide attachment on the surfaces of the cell plasma membranes that helps in the cell recognition phenomenon. Main role of carbohydrate in the plasma membrane is the cell recognition phenomenon.
Sir, C. D.
C.
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 Yes. It is the nucleus. It is the largest cellular organelle. Nucleus is the largest cellular organelles which can be visualized with the light microscope. Of course, in some of the powerful microscope, means light microscope itself, mitochondria also to some extent is visible. But if there is option nucleus and mitochondria, then you have to go for nucleus. Otherwise, if nucleus is not there, if it is a mitochondria, then you have to go for mitochondria. Okay. Otherwise, if both nucleus and mitochondria, you have to go for the nucleus. Nucleus is the largest cellular organelles, always visible with the light microscope. These are the largest cellular organelles.
So B.
Sir, D. D.
B.
Any option? Yes. Which one?
C.
Size and shape of mitochondria is varies in the cell. Mitochondria can fuse with one another. Large mitochondria in the cell can split into two because it is semi-autonomous organelles. In one cell, one mitochondrion will be exceptionally larger than others. It is the D, it cannot be. Yes, the size and shape may varies, but in one cell, one mitochondria, okay, cannot be exceptionally larger than others. It is an answer D. In one cell, one mitochondria cannot be exceptionally larger than the others. Okay, answer is D.
Sir, why not B?
Mitochondria in a cell can fuse with one another. It can fuse with one another.
Sir, how mitochondria divide by binary fission?
Yes. It can fuse with one another. It can fuse also. Mitochondria can fuse also.
Okay, sir.
Sir, A.
It is
A.
lipoprotein, golden brown pigment stainable with certain fat dyes that is commonly found in cardiac muscle, liver, and some nerves. So it is the A.
Sir, B.
D.
Sir, B.
Tell me nicely.
The slide is not visible.
Slide is not visible. Just a minute. Is it visible?
Yes, sir.
Yes, sir.
So what is the answer?
Sir, B. Autophagy.
Yes. When the cellular organelles become obsolete, they are disposed to process during which their endoplasmic development is the autophagy. If the damage or the excess cellular organelles are disposed, it is called as clinophagy. Okay. It become obsolete. Okay. Then it is the autophagy. Huh? Outdated. Done. Their work is done. They have done the functions and become obsolete. And heterophagy means exogenous RBCs, the microorganism, other things. Okay, and it is the heterophagy. Yes, colchicine arrest the mitosis. They prevent the mitotic spindle formation.
Sir, exocytosis. A.
Yes. Correct answer is exocytosis. Extrude its content, exocytosis, outside. Endocytosis, evaginations.
B.
Huh?
B.
You justify your answer. Justify your answer. Polar N means?
Hydrophilic.
Non-polar?
Hydrophobic.
Tail. So tail means they will be at the center. So peripheral membrane protein will be at the periphery, is not it? So they are at the periphery, at the surface. It is at the polar end of the phospholipid, is not it? Correct? Understand? So it is the A. Answer is the A. Peripheral membrane proteins of plasma lemma remain in contact with polar end of phospholipids. Okay? A. So here already I told this answer.
D.
Yeah. No, here D. Is not it?
Yes, sir.
Yes, sir.
Yeah, alright, so answer. So that's all for today. Okay?
Yes, sir.
So
Thank you.
Thank you, sir.
this is about my topic, and just a minute. Now the next slide. Next you will be dealing with the tissues of the body. Okay? So there are 4 basic tissues in our body. So entire our body is made up of, because the cells will form the tissues of the similar cells of the similar functions they will perform, and they form the tissues. And in our body you will find that there are 4 basic tissues of the body, that is called as the epithelial tissue or the epithelium, connective and supportive tissues, the muscular tissues, and the nervous tissues. Okay? So the epithelium or the epithelial tissue, connective and supportive tissues, muscular and the nervous tissues. So the next topic will be covered up, I think, with Dr. A.D. Patel. He will deal with the connective and supportive tissues. Then on teachers from Jalukri, Dr. Kima will deal with the epithelium or the epithelial tissue. Then from our Dr. Orup and Dole will take part in the muscular and nervous tissues. Clear? So these are the 4 basic tissues which forms our entire body. And after studying these 4 basic tissues in between, in a normal class,
Yes, sir.
we will go to the spatial histology, the structure, the organization of the organs. So, but before that, you must know what are the basic tissues of the body. Our entire body is made up of epithelial tissue, connective and supportive tissue, muscle tissue, and nervous tissue. With these 4 tissues only, whole our entire body system is formed. Okay, but in which organ, what are the tissues you will find, in what ways they are there, it is varies in different. But these are the 4 tissues you will find in our entire body. Clear?
Yes, sir.
Next Saturday, your class will not be there, being the second Saturday holiday, but next to next Saturday it will be deal with. Okay?
Yes, sir.
Thank you, sir.
Yeah.
Thank you.
Yeah, most welcome.
Thank you, sir.
Thank you, sir. Thank you, sir.
I will give you the text. I will give you the text after some time.
Thank you, sir.
Thank you, sir.